Endocytosis, as you probably all know, is the process by which the cell takes in stuff, such as nutrients and ions, etc. The things come in and form what's called an endosome. If we zoom in here, we will look at the three types of endocytosis. We will go over them briefly one by one. First one is basic phagocytosis, usually involves a bacteria or a virus. Here is the intracellular fluid. On the outside, there are many ions and proteins and other things as well. Phagocytosis is basically when the cell detects the presence of a bacteria and tries to engulf it. It phagocytizes it, and through eating it, it will form an endosome. The other form of endocytosis is bulk endocytosis, which is where the cell needs to take in some water or something to expand its volume. As it's doing this, it takes in some ions as well, and also it forms an endosome, like so. Now the last type is what's called the receptor-mediated endocytosis, which is where the cell's receptor detects cargo molecules on the outside, and through the help of adaptins, which helps in specificity of the cargo, the cell can take in the cargo molecule. In this case, a protein is detected by the receptor. The membrane then forms what's called a coated pit, where it slowly invaginates the substance as well as the receptor and adaptin, forming an endosome. We will focus mainly on the receptor-mediated endocytosis later on. So now, going over the three types of endocytosis again. Here we have the phagocytosis, bulk endocytosis, and receptor-mediated endocytosis. Each of them will form an endosome, which will later form an early endosome, where all the endosomes brought in will combine with each other. Now this early endosome has a few fates, one of which that it can be recycled back into the plasmalemma to expand the cell membrane, or the early endosome can turn into a late endosome, which can later evolve into a lysosome. So now let's begin focusing more on the receptor-mediated endocytosis. But before that, let's learn some key words. First is the protein known as clathrin, which is a triskelion, adaptin, which helps in cargo specificity, accessory proteins, which help in taking the cargo in by recruiting clathrin protein, and finally dynamin, which is a protein which pinches off the endosome being formed from the membrane. So here we have the membrane with the cargo receptors and the adaptin attached on the inner membrane part of the receptor. And here we have the cargo. On the inside of the cell, we have lots of clathrin proteins. The cargo will bind to the cargo receptor, which will then cause the adaptin, A2 is the most common adaptin in humans, to recruit accessory proteins, which will then help in recruiting clathrin protein. So the clathrin will start binding with each other to form hexagonal type shapes, things, which will eventually pull part of the membrane in, forming a coated pit, or a clathrin pit. More clathrin are still recruited and it just gets built up. This again causes the membrane to sink partially in, taking in the receptor and all that in that segment of the membrane. The clathrin protein continues to attach with each other, slowly forming a sphere-like structure. Dynamin also begin to attach to where the endosome is being formed. Finally, when the vesicle is ready to be pinched off, dynamin will form a helical structure around it, and using energy from GTP, dynamin will break off the endosome, freeing it into the cytosol. Now, as clathrin-coated vesicle is moving in the cell, clathrin will start breaking off, going back into the cytosol, leaving a naked vesicle. So as you can see, clathrins are breaking off, detaching, leaving a naked vesicle. This will allow the vesicle to fuse with the early endosome freely. And as we discussed earlier, the early endosome has a few fates. One is that it can be recycled back into the cell membrane, or it can turn into a late endosome as it proceeds towards the center of the cell, where it can then evolve into a lysosome with the help of the Golgi apparatus. I hope that all made sense to you. I hope that wasn't too quick. Please comment, like, and subscribe, perhaps. Thank you.